First of all, I want to thank for the opportunity to speak here at PEROSH. As I've been introduced, my name is Solveig Krapf, and I'm a postdoc at STAMI, the STAMI gang, the National Institute of Occupational Health in Norway. And my talk is entitled Mycotoxins, Where Do We Find Them and Why Are They Important? So this is a project where we try and close the data gaps on the natural toxins' effect on human health. And this project is a part of PARC, the Partnership for the Assessment of Chemicals, which was mentioned earlier. And this was established after the European Commission adopted its chemical strategy for sustainability in 2020. And the main objectives of PARC is to promote innovation of safe and sustainable chemicals, which is very important for us working in occupational health with occupational settings. And for traditional toxicity test methods, they are very time-consuming and costly. They require a very high number of test animals, and there's always the question of relevance. So if we consider the iceberg of chemicals available on the market, the part of the iceberg above water is the part of the chemicals that we actually know something about. But knowing icebergs, there's always a larger chunk underwater where our knowledge is still lacking. And then I haven't even mentioned the natural toxins, which perhaps are not commercial available. Toxins naturally occurring in certain foods, like fruit, veggies, legumes, and fish, or mycotoxins, which are produced by molds. And this is what we will be working on. So the goals of PARC is to provide data to fill the knowledge gaps of poorly characterized contaminants or emerging hazards, such as mycotoxins, contribute to the consideration of new approaches and methods of health assessments away from animal testing, by promoting the use of innovative methods and tools to contribute to the integration of new technologies. But let's move back to the mycotoxins. Where do we find them and why are they important? So, as mentioned, mycotoxins are metabolized, produced by different fungi or molds, and molds of the genus Alternaria are ubiquitous plant pathogens and saprophytes, which can adapt to a wide range of temperatures and humidity. So they're literally found all over the globe. And the Alternaria molds is well known to contaminate a broad range of crops and raw materials. And while visible infected foods usually are avoided, the mycotoxins are not visible, however, can be in and around the area of the contaminated plant. So the toxins are also found in processed versions of the contaminated food, which are foods that many of us consume daily, such as cereals, cooking oils, apple juice, beer, wine, and so on. So we're all exposed to this in our daily life. Due to a very limited toxicity data available, a threshold of toxicological concern approach has been used to assess the risk of mycotoxins. But in addition to the dietary exposure to mycotoxins, by intake of contaminated food, humans are also exposed in occupational settings. And biomonitoring studies of multiple mycotoxins, including this Alternaria toxin, in the airborne and settled dust in occupational environments throughout Europe, Asia, and Africa, have shown that both farming, grain handling, waste handling, as well as working in bakeries and in the food production represent exposure risks. That's mainly through the inhalational and the dermal route. And limits for the total daily oral intake in humans have been established based on risk assessment, but they're not considering the additional risk from occupational exposure for these employees. But why do we really care? Why are these mycotoxins important? Well, as the name suggests, they are toxic. There's found evidence that Alternaria can be cytotoxic, where the exposure leads to cell damage or death. They can be genotoxic or mutagenic, where the effect is on the genetic material, leading to cancer. They also are endocrine disruptive, sometimes mimicking hormones and potentially have an effect on the reproductive system, or immunotoxic and immunomodulating, where exposure can have effect on our ability to fight other diseases. So different partners will be working on deciphering these different toxic pathways. And at STAMI, we will be working on the immunotoxic or immunomodulating effect in our experiments. So we're going to use a cell that is engineered to mimic certain factors of the immune system. And if we treat this cell with a substance that typically invoke an immune response, there will be produced a color signal, which can be read in a spectrophotometer. So we want to use this model to explore how Alternaria toxins invoke an immune response, and also in combination with material from other microorganisms, such as lipopolysaccharides, LPS, from bacteria. So to explore the potential combination and modulating effects. So this will give us a background understanding in the interplay between the toxins and certain parts of the immune reaction. And then we will go on to using a inhalational cell model. And as this is one of the two most common routes for exposure in occupational settings. And when you want to mimic the exposure through the lungs, there's two things to consider. The first thing is how the lung exposure occur, which is through breathing. And to mimic this, we will use a cloud system. We'll be adding the mycotoxins at the top there, and it will be dispersed into a cloud, which then comes evenly on the cells, like similar to breathing. The second thing to consider in an inhalational model is the biology of the lung. And to mimic this, we will be using the commercial epi-airway, which is a co-culture of cells. And these cells are also grown in the interface between air and liquid, which are mimicking the air-blood barrier in the lung. And then after all this exposure, we will be looking into the gene expression to try and understand how the mycotoxins affect the immune system. So what is it that we want to achieve? Well, we want to contribute to the PARC goals. We want to try and close the data gaps in the knowledge of the mycotoxins by understanding their immunomodulating effects, and hopefully in a faster, cheaper, with reduced animal testing and more relevant way. And of course, ultimately, that this leads to improving occupational health settings and new exposure limits. And as you can see, there's a lot of institutes in PARC which are involved in these natural toxin work, looking at it from different views. And it's very important that we also in occupational health take part of this, as there's by now too little knowledge and actually a lot of exposure. That was all for me. Thank you.
Yeah, thank you very much, Solveig, for your excellent presentation. I learned a lot because I didn't know so much facts about microtoxins. It's just a question if we can't see it, maybe in fruits or in other nutrition. Do we have, as humans, a certain warning system by taste or smelling them?
I don't think so. No,
okay.
no, not for these. Like the mold, you will see, right?
Yeah.
But even if you remove the one moldy fruit, it could be the toxins are.
Okay.
Yeah. So that's how they, I guess, end up in the processed versions of the foods, because even though they remove it, they don't get rid of these things.
So very dangerous subjects.
Very dangerous.